All right, so let's move on to finish off our grand tour, and we're going to end it all with talking about chemistry. How do different parts of the systems work together? We talked about looking at the different kinds of levels of molecular biology, DNA, RNA, protein, and looking for patterns in those. What I want to do is I want to say, as a chemist, I want to say, are there different chemical patterns that we can do the same thing with? How does chemistry interact with biology? And this actually has something to do with your chemistry biology lectures, which will be part 3, presentation 3, that you're going to be giving, lectures that you're going to be giving later in the class. So the first topic I want to talk about is there are some ways in which metals affect immunity, and you can talk about different branches of immunity. Remember that you have the innate fast immunity and the slower but more adaptive immunity of the T cells and B cells. And you can talk about how zinc, iron, and copper interact with those. Here's a review about that, and it has a lot of detail. I'm just going to say that this review is out there, and people are looking at these patterns. Here's just some figures from the review. It talks about how zinc works. Zinc is a regulated molecule. It's pumped in and out, and it's actually used by the cell for certain kinds of signals. Are there certain kinds of immune signals that like to use zinc? There are certain kinds of ways that the zinc will activate, and there's some antiviral immunity that is actually, zinc plays a major role in the signaling for it. And this is one of the reasons why I think zinc supplements are actually good to take, because if your immune system depends on zinc for signaling, let's give it some more zinc so that it can fight off against the cells and it can have the zinc that it needs to send these signals right here. By the way, if you want to look in more detail, you can see that, for example, zinc is involved with blocking calmodulin. You can get to the Krebs, and you see down in the corner interferon gamma. That's one of the major immune signaling molecules that we talk about. So zinc is associated with that. Then they have a figure about iron. Iron, on the other hand, is not used the same way as zinc because it has a different role in natural history. You can even come up with some ideas about why this might be. Iron will actually go inside the cells, and it will actually be pumped out of the cells. Usually, we want to keep the iron concentrations, the free iron concentrations, low inside the cells. But the iron does actually activate some kinds of cell responses by making ROS. It's a little bit hard to see, but the little green blotch way down there in the left corner, that says ROS. And through that ROS response, the iron actually induces certain kinds of responses from the cell, which can be protective and can be antiviral. Again, more details can be found by looking in this, but I just want that to make sense to you because iron has a particular role. It looks different than zinc. And copper looks different than the other two. Copper actually has direct antiviral activity. It also generates reactive oxygen species, but it results in a different pattern of reactive oxygen species generation. And I believe, if I'm reading this right, that inside the nucleus you actually have more of those patterns being made. And so you have the proteins that are induced by copper are going to be different than the proteins that are induced by others, and ROSs are at the middle of it. And remember, those are also at the middle of natural history. So that's the first topic, but of course even this leads to the main actor in A World from Dust, which is oxygen. And I just want to say that oxygen has a major central role in immunity. I'd be curious if you could find a paper about how that might have evolved. Oxygen might have resulted in certain aspects of immunity evolving. I have some hypotheses, but I'd like to know if you found a paper on that. That could be a good chemistry and biology paper. Ask me, and I'll see if it can work. This is a paper that wouldn't work for that because it's a review, but it does point to several experimental papers that would be in that area. And the whole idea is that reactive oxygen species cause damage. And there's a whole hypothesis in the immune system where one of the primary drivers of immunity is the fact that we've been damaged at some point. And the immune system is responding to the cell damage first and foremost, and then everything else sort of comes around that. There's pluses and minuses to that, but one of the strong points is that it does have a central role for chemistry because it has a central role for damage. The other thing about this, I don't know if you picked up on this by scanning the words, but this is about immunity, but it's only about C. elegans. It's about worm immunity. This is not in higher levels yet. But they do have this really nice chemical graph. Remember that we have the immunity is about finding the middle of the response. If you have too little response, you have deficiency. Too much response, you have autoimmunity. And the top is saying you want to be right in the middle. What they're saying with this is they think damage is the main driver of this axis. And you want the damage to be in the middle, where you're doing enough damage to the bad things, but not too much damage to the good things. Because damage is related to oxygen and ROS, they say that the damage and the ROS go together. And in fact, you have this idea that over time, you have as you have ROSs going up, an infection will involve ROSs going up because of the damage, and the cell detects that, and the cell even uses ROS to cause damage in the way it wants to at some point. If you have something getting out of control as you move to the right, you can have the ROS level suddenly shooting up. You have a redox buffer capacity, and A World from Dust talked about a bit of this, that you have some ROS buffers, oxidation buffers in your cell. When those get overwhelmed, then you cross over the line into the bad part of the graph, and you start to shoot up into the case where you are starting to be, the redox is starting to damage your cell. And so this is all about C. elegans, and it's a very crude model of immunity, but they're doing that. I want you to sort of look at this and maybe fill in the blanks for what this is. Hallmarks of intestinal pathogenesis. So what are they looking at in C. elegans that is causing the damage that they are measuring? And the first one is mitochondrial damage, and you see that word over and over again in the paragraph that follows it. Mitochondrial damage there, mitochondrial damage there, and there are bacteria that cause the mitochondria to be damaged, and that's ROS mediated. Then not just the mitochondria, but the intestines are destroyed. That's number 2 on this, and there are those words about intestinal destruction, here, there, and there, where they're talking about there's a certain bacillus that infects C. elegans, and then you see damage resulting, maybe not just from the bacillus, but also from the immune response in the C. elegans intestine. The third way that they see ROS, the third way that they see all this working out in C. elegans is actually that it doesn't get enough food, nutritional deficiency. And they see ROS involved in that as well. And of course, you got to remember, immunity is cells growing. It's energy being spent. You need to have good nutrition to have good immunity. And so they're talking about the nutritional content of the bacteria they ingest can be connected to the ROSs that are signaling infection, and nutritional change could be playing another role. So mitochondrial damage, intestinal destruction, and nutritional deficiency are the 3 connections that they make between ROS and immunity. And so a lot of times ROS can be produced deliberately, or it can be produced accidentally in the cases of immunity. So they have, by the host, they say that the C. elegans can produce ROS to fight off things. It has a particular oxidase that is activated. I'm curious if that oxidase has a metal in it, but they don't say. We'd have to look it up. There can be ROSs that are generated when the bacteria damage the host. So there's the C. elegans making the ROS. There's the bacteria damaging the host with the ROS. And then that's the bottom by the pathogen, by the way. But then there's the when the mitochondria themselves are damaged, they will leak ROS. So that's a third source of ROS production. They had this big, huge diagram, and I'm not going to go through it in general except to show you how a diagram like this can be taken apart. You just want to take it step by step. And what's the key to this is that in the middle of this diagram, I had to break it into 2 halves, so it's at the bottom of this one, you have the ROS that are at the middle of everything. You have all these other things around it. So you have number 1, which is the pathogenic bacteria are actually infecting them, and they're sensed by chemosensory neurons that turn on this oxidase. This BLI3 DUOX oxidase is a key thing to take away from this review. If I was reporting on this, this is what I would focus on. I would actually focus less on all the other things. That's why they put it as number 1. But then they also have the nutritional deficiency, so the oxidase is deliberately turned on by the bacteria. And that makes ROSs. There's also nutritional deficiency, which causes low ATP, which it relates to ROS through that. And then you have number 3, don't let me miss number 3, the mitochondria themselves can be damaged and they can leak ROS. And that's how you can get elevated levels of ROS in infection. Basically, there's lots of ways you can get ROS in infection. You can think about, from the story of A World from Dust, why does ROS result from all of these things? You move down to the bottom of the figure, and you see the cellular proteins that respond more specifically. Number 4 is about the ROS causes damage to the structures of the cell. And so that's just sort of the biochemistry of reactive oxygen. That has to do with how reactive oxygen species can react. And then you have, when those things are damaged, the cell turns certain proteins on. So you have these transcription factors that are made. There's this long list right here. And some of these specific things might be interesting to look at in isolation. But together they say there's a lot of genes. These specific genes are turned on by the ROS that result in antimicrobials being made that go back and attack the antigen, the bacterium that produced things in the first place. And there's also cytoprotective proteins that are made. Things that will make antioxidants are the obvious one here. The cell will make antioxidants to fight against the high levels of ROS. It will also use those ROS to signal. So it sends a good signal, but it's also damaging on the other hand. You can see how you have a complex system with a balance in the middle. Then there's number 6, which is where the proteins that are made for cytoprotection, how do they actually protect? In some cases, they block the ROS. In some cases, they block the damage to proteins. I guess they can block both at the same time. But there's lots of ways about how are the cytoprotective things cytoprotective. And then finally, what about when it's just too much, when you cross over the line, too much ROS, and the cells are destroyed? There's one interesting key idea here. The key idea is that the cells are overwhelmed first, before the organism is. So the cells begin to malfunction and die first from the ROS. Exactly which cells must die for the organism to perish is not known. So they're saying that this is an active area of research. Maybe there are cells that are more sensitive to ROS, and maybe there's a reason for that. So the chemical way of looking at things might actually give you an idea of why are certain cells dying first and resulting in the catastrophic organismal failure. So all this is put together the same way. Remember, they have those long lists of proteins that are transcribed in response to ROS? Well, this spells out for each protein what they know about it. Really complex, lots of overlapping arrows. But the main thing I want you to see, ROS at the top, and then for each individual protein, you can see which individual things it produces to protect the cell. Some of them only make antimicrobials. Some of them only interact with catabolism. And it's really complex, and that's why sometimes a computer helps with all this. But you can look for the things, and the main things are, you can look at these individual things. The thing that stands out to me, look right in the middle. HIF1 alpha. That means hypoxia-inducible factor. Hypoxia means low oxygen. Oxygen is at the middle of all of these things. What does that mean, and how can you connect that to the chemistry of what oxygen is? And that's how you can present these complex chem bio papers. Focus on the elements that we have in common with A World from Dust and build out from those, because for all of those elements, you have a story of how that fits into natural history. And from that, you can say oxygen's a double-edged sword. Here it is, being double-edged. And it's used for these factors. The hypoxia-induced factor makes antioxidants. And that all fits together if you look at it from the standpoint of oxygen. This is one point at the end of the paper where they come out and say, hey, the one thing that we think is really important is BLI3-DUOX. So when you're faced with a paper like this, usually there's one or two things that they're trying to get you to come away with. Look for those things where they repeat it multiple times in a paragraph, and then focus on that paragraph. You don't have to know every paragraph of a paper. You have to find the key ideas. Key idea is right here for this paper. And, of course, the big question, the big drawback to this, you can see it from the beginning. They're dealing with worms. We are mammals. Does this really work for us? Well, here they argue, we think it does, for this reason, but there are these problems as well. But here are where we think it overlaps most. with particular interleukins, with particular receptors, this TLR, and there's also the particular ROS that's generated by NADPH oxidase or generated by mitochondria. We certainly have mitochondria that can leak ROS, so at least that part of it definitely can apply to mammals. It's an interesting question. How much does it apply? Do we have sturdier mitochondria? That's the kind of questions you can ask when faced with a paper like this, and I want you to bring the chemistry and the biology together, especially for presentation 3. That's one of the reasons why I end up with this. I like this review because they say key open questions. Just remember, you're not going to be presenting a review, but you're going to be taking a couple papers. Like I could take a couple papers from this. I would focus on the BLI 3, and then I would actually try to expand those papers if I wanted to go in that direction. Needless to say, you're not asked to pick a paper for this, but this might help you select a paper for presentation 3. And if you do find a paper that you want to do that you think might fit, give it a try. Give me an ask, and I'll see if I think it will work or not. All right, and that is all we have to say about structural immunology. Thank you for hanging in there, and I will see you at the next class.